everybody, so in this video we're going to be talking about B cells and T cells and how they undergo maturation, specifically how B cells and T helper cells go from naive lymphocytes into mature lymphocytes and are able to elicit a really specific immune response to a particular pathogen or antigen. Okay, so we're going to start on the B cell side of things. And just as a side note, B cells and T helper cells kind of, undergo activation through the same process. And what I mean by that is that they go through these same 3 steps. And those steps are antigen recognition, co-stimulation, and cytokine recognition as well. So I went ahead and put the steps here just for your reference, and we're going to go through each one of them. And like I said, the steps are the same, but the way they undergo those steps are slightly different. So we're going to go through each one separately. And I'm going to go ahead and start with B cells. So B cells are a little bit different from T cells in that they are antigen presenting cells themselves. So whenever they see an antigen, so I'm going to go ahead and draw my B cell here. So I have a B cell, and on the surface of that B cell, it has a BCR, which is just an immune globulin, and specifically a naive B cell, which is what we got here, is going to have IgM. So it's a BCR, and that BCR can recognize a particular pathogen. So I'm going to go ahead and draw my pathogen here. And, I'm not the best drawer in the world, but I think bacteria is pretty easy to show. It might look like a peanut to you, but to me it looks like a bacteria. And that bacteria is going to stimulate that B cell receptor, and then that B cell is an antigen presenting cell, right? So it can present that antigen on the surface of an MHC class 2. So essentially it recognizes the antigen, and then it produces it on its surface. And now this is where the B cell needs a little help. The way I like to think about it is B cells aren't very smart. They, can recognize like, hey, this is something, but they don't know whether it's dangerous. They don't know if they need to do something about it. So they need help from a T helper cell. So I'm going to go ahead and draw my T helper cell here. And then on the surface of the T helper cell, we have a TCR, which is a T cell receptor, just as the BCR is the B cell receptor. And that TCR is going to recognize that antigen on the surface of the BCR or the B cell in the MHC class 2. And of course, this is a very specific T cell, specific TCR for that antigen MHC. It's not just any T cell. It just is by chance it runs into a T cell that can recognize this antigen. And that is what we call signal number 1. And that is our antigen recognition step. So this one right here. Okay, and so what is the next step? So now, the T cell says, hey, I see that you have something. Now the T cell needs to either say it's dangerous or it's not dangerous. And the way it tells the B cell if that is dangerous is through a ligand called CD40 ligand. So it's going to upregulate this ligand. So obviously it would upregulate in this case because we're dealing with a bacteria, so it knows it's dangerous. So CD40 ligand is going to be upregulated, and then on the surface of the B cell, we have a receptor called CD40. So pretty easy, right? So CD40 receptor is going to see CD40 ligand, and that is what we call signal 2. And we also call that co-stimulation. And that co-stimulation is what allows the B cell to know that there's danger, that, hey, B cell, you should probably do something about that bacteria, because otherwise it doesn't know. It just says, hey, I see something, but I don't know if it's dangerous or not. Okay, and then the last signal is cytokines. And what happens here is that we have our T cell over here. It's going to produce cytokines. These are just molecules they're signaling molecules essentially that tell cells to do things. That's what cytokines are. So this T helper cell over here is going to produce cytokines based on what we're dealing with. So it sees that we have this bacteria, right? So it knows it's a bacteria, and so it's going to produce specific cytokines that tell this B cell, hey, we're dealing with this specific bacteria, you need to do this specific thing. Okay, so then what happens? So now we have our 3 steps. Oh, and by the way, this is our signal 3, right? And that's our cytokine production. So now we've gone through all 3 steps, signal 1, signal 2, and signal 3. Now our naive B cell can become a mature B cell. And the way that this happens is through 2 things. So our B cell, it's going to come down here. And again, this is IgM on its surface. It's going to go through first affinity maturation. And our B cell's activated, right? And then once the B cell goes through affinity maturation, which essentially what this is it just makes the BCR stronger. So we're just trying to make that B cell receptor stronger, more specific, and better for this specific pathogen that we're working with. And then once it goes through that, it goes through class switching, which, as I mentioned previously, we have IgM on the surface, but in order to be really specific for, whatever pathogen that we're working with, we want to be able to produce IgG, IgE, IgA, in order to make the immune response more specific. And of course, we're going to be able to make plasma cells that secrete these immune globulins, and we're going to be able to make more B cells that have these on the surface so we can better recognize the antigen and become, just overall elicit a better immune response. That's what we're trying to do here. Okay, so T cells are pretty similar. Again, they're going through these same 3 steps, but it's just a little different in how they do it. So T cell, just like the B cell, is going to be naive when it first starts out. So naive meaning that it has the TCR, it can recognize an antigen in the context of an MHC class 2, but it isn't specific. It's just a T helper cell. It hasn't gone into a specific type of T helper cell yet, so it just is generally out there waiting to be activated so it can be more specific to the immune response. And I'm just going to go ahead and start with our end goals here because I think that makes it a little bit more concise and less confusing. So yeah, we're going to go through all 3 of those steps, but our end goal here is we're going to want to produce Th1, Th2, Th17, or follicular T helper cells. And essentially these are just specific types of T helper cells that are for different things. So for example, Th1 helper cells, so T helper cells type 1, are best for fighting intracellular pathogens. And whenever I think of intracellular pathogens, I'm really thinking about viruses is the main example. Th2 are what we use to treat surface pathogens. And really, when I think about those, I'm thinking about big parasites like helminth worms, roundworms, stuff like that. Th17 is extracellular pathogens. And that is, bacteria and then also fungal infections. And follicular T helper cells, we're not going to go into a bunch of detail, but they're in the follicles of the lymph nodes and they help differentiation for B cells. So we will cover that in a different video. Okay, so going back to T cells, how do they get here? How do they get to Th1, Th2, Th17, and follicular T helper cells? Well, they do this through those 3 steps: antigen recognition, co-stimulation, cytokines, to tell them which route they need to go in their maturity. So T cells are a little different in that they can't recognize antigens themselves, right? So they need an antigen presenting cell. And it can be multiple different things. I'm going to go ahead and use a macrophage as an example, but just keep in mind the antigen presenting cells, they can be macrophages, they can be dendritic cells, and they can even be B cells, as we've seen previously. But here we're going to use a macrophage. So on the surface of this macrophage, we have a PRR receptor. A pathogen recognition receptor is what that stands for. And it can recognize that pathogen. So I'm going to draw my bacteria again. So it recognizes that bacteria, and it can, just like the B cell could, upregulate it on MHC class 2 molecules. And now our T helper cell can go on to recognize that antigen, because T helper cells can only recognize antigen in the presence of MHC2. They can't recognize it in the context, like on the direct pathogen. They wouldn't be able to do that, like macrophages, dendritic cells, and B cells can. So they recognize it through MHC2 on the macrophage. And then we need our co-stimulation. So this would be signal 1, which is antigen recognition, but now we need co-stimulation. And the difference being is it's not the T helper cell stimulating the macrophage. In this case, it's actually the macrophage that's going to stimulate the T cell and tell it, hey, you need to differentiate, you need to become mature, you need to be more specific to this immune response so we can do better. And the way the macrophage and the other dendritic cells will end up doing this is through upregulating something called CD80 and CD86. So on the surface here, we have CD80 and also CD86. And both of those are going to kind of do the same thing, and they're going to be recognized on the surface of the T cell with a receptor called CD28. So CD28 is going to recognize CD80 and 86, and that is what we're going to call signal 2, which is co-stimulation. So this is telling the T cell, hey, this is dangerous. You need to do something about that. And likewise, the macrophage is going to be producing a bunch of cytokines that say, hey, this is a bacteria. So they're going to say, hey, this is a bacteria, and you should do something that's specific for bacteria. So this is our signal 3, which is cytokine recognition. And those cytokines are going to tell that T cell, hey, this is exactly what you need to do to fight that dangerous response. And in the case of this, since I drew a bacteria, we know that this cell will become a Th17 specific. And then that Th17, along with really all Th1, Th2, and the follicular T helper cells, are going to be producing cytokines. And specific cytokines depending on if it's Th17, Th2, and so forth. And those cytokines are going to help other immune cells. And they help other immune cells to do their jobs better. So essentially that's what T helper cells do, right? We're just trying to help other immune cells. It's in the name. And I'm just going to zoom back out so you guys can kind of see the whole big picture again. I hope that this cleared things up and made things a little bit easier to understand. If you have any questions, please feel free to comment. Please like, subscribe, and thank you guys for watching.